Hello friends, welcome to VMH Learning. This video is about radial immunodiffusion. Radial immunodiffusion is also called as Mansini immunodiffusion, sometimes also referred as single radial immunodiffusion assay. So how radial immunodiffusion is useful to us? It's an immunodiffusion technique used in immunology to determine the quantity or concentration of an antigen in a sample. Radial immunodiffusion is a type of precipitation action. That means it is based on the principles of the precipitation curve. And the principles of the precipitation curve states that interactions of antigen and antibody with each other form visible cross-linked precipitate when the proper ratio of the antigen to antibody is present. Let's understand this concept with the procedure of radial immunodiffusion. First of all, we make a solution of agar. The agar solution contains an antibody, say immunoglobulin G. Agar solution with antibody is poured into a glass plate. The solution is poured in a manner that it makes a uniform layer onto the glass plate. Carefully circular wells are cut into the agar. A single or series of standards containing known concentration of antigen are placed in separate wells, while control and unknown samples are placed in other remaining wells. The different concentrations are shown as well. Specific antigens to the impregnated antibodies diffuse through the agar in all directions from the well. Diffused antigens react with the antibody incorporated in agar gel. The interactions between antigen and antibody form visible precipitates or precipitate rings in the area of optimal antigen-antibody concentration. Ring-shaped bands of precipitates forming concentrically around the well indicate immunological reaction. The diameter of the precipitate ring formed corresponds to the amount of antigen in the solution. The ring diameters are measured and noted. Finally, a standard curve is prepared using the ring diameters of the standards versus their concentrations. This curve is then used to determine the concentration of the control and unknown samples. Now let's discuss about the result interpretation of radial immunodiffusion. The presence of a precipitant ring around the antigen wells indicates specific antigen-antibody interaction. Absence of precipitant ring suggests absence of reaction. The greater the amount of antigen in the well, the farther the ring will form from the well. Now there are many applications of radial immunodiffusion. This is used to determine relative concentrations of antibodies in serum. We can estimate serum transferrin and alpha ferroprotein with this technique. This technique is a very powerful tool to compare properties of two different antigens. It's also used to determine the relative purity of an antigen preparation. The technique is employed for diagnosis of different diseases. We also use this method in serological surveys. This was all. Thanks for watching.